Hi, my name is Dedi Meiri. I'm a scientist. I'm a professor in the Technion, which is the Technology Institute of Israel. I'm running a laboratory for cancer and cannabinoid research, even though in the last years we're doing a lot more around cannabis and research in general in other areas. Background in biochemistry, master in biochemistry, I have a PhD in molecular and genetic plant biology. So I'm coming with a strong plant background. I'm a son of agriculture. I grow in the fields. I was for 7 years the head of the botanic gardens in Tel Aviv University and the medicinal plants garden. Moved for 4 years to do postdoc in cancer research in Toronto. And in the last few years, I'm merging these fields, my knowledge in plant and my knowledge in cell biology, cancer biology, neurobiology, to bring them together to higher level. Cannabis is a great opportunity to merge this together.
The Curious About Cannabis podcast is produced by Natural Learning Enterprises, a mission-driven company dedicated to enhancing critical thinking skills and public scientific literacy about life in the natural world. If you like Curious About Cannabis, consider checking out some of these other learning initiatives by Natural Learning Enterprises.
Come on, Molly! It'll be an adventure! Phoebe called out as she followed Brother Toadstool. Brother Toadstool led Phoebe and Molly into a tunnel that went deep down into the ground. As they climbed into the tunnel, they found themselves getting smaller and smaller and smaller.
Our new children's book, A Toadstool's Treasures, takes young readers on an adventure into the fun and fascinating world of fungi. Learn more and find mycology-related learning resources, games, and lesson plans for teachers and homeschooling families at toadstoolstreasures.com. Biodiversity loss due to habitat loss and fragmentation is rapidly increasing around the world with devastating consequences. Learn how you can help contribute to native habitat corridors in your community and reconnect with your wild neighbors at gardenwild.org. Oregon recently became the first state in the United States to legalize the medical use of psilocybin. As cities all over the country begin to decriminalize the use of entheogenic plants and fungi, it's time to have a serious discussion about psychedelics. The Serious About Psychedelics limited series podcast is coming soon. Learn more at seriousaboutpsychedelics.com. You can learn more about Natural Learning Enterprises at naturaledu.com. And now, back to the show. Hey everybody, this is Jason Wilson with the Curious About Cannabis podcast. Thanks so much for tuning in once again. So today I am beyond excited to be talking to the guest that we have today. I'm here with Dr. Dedi Meiri from Israel. If you've heard me talk at length about cannabinoid science and research, you've heard me mention Dr. Meiri and his team's work. Welcome, Dr. Meiri. Thanks so much for being willing to come on the podcast and chat with me today about cannabinoid science.
Thank you for having me. It's a pleasure.
Most excellent. And to really kick this right off, because I've got plenty I want to dive into, do you mind sharing a little bit about the lab that you're working in now and some of the, focuses of your research up until now, and then we'll kind of spin off into all sorts of areas of things that you're currently studying?
Yeah, so my lab, in 90% of my lab working with cannabis. I have another few projects in the lab, but most of the lab is working around cannabis. When we started 6 years ago, we want to see how cannabis affecting cancer cells, the anti-cancer properties of cannabis. We know that patients use cannabis treating the palliative treatment of cancer, so improving their quality of life. the cancer patient, improving appetite, mood, reducing nausea, reducing pain. But we want to ask whether cannabis can also be uh, with anti-cancer properties. We laid on data and research that come from Manuel Guzman and Christine Sanchez, but we all know also about Rick Simpson and other things. And the question was, is it true that cannabis can be anti-cancer? And when we started to work, we were quite shocked. I was quite shocked about the knowledge and the tools that I have to do this research. I'm coming from very accurate, modern type of research, and this was 100 years back. So the reason that there was illegal to do research around that, and there are other reasons, and also the regulation. When I applied to do cannabis research to the Minister of Health in Israel, cannabis is under the healthcare from 2007. So it was 2014. I was applying for doing cannabis research, and I got day after that a license to work with cannabis. And my license is R for research 0001. So you understand how much collaborations I had that day. So I was the first one to apply. It's true that in Israel we have Rafi Meshulam, that I'm sure if people are in the field of cannabis, they know his name. He was 93 weeks ago. And I was interviewing, I'm a good friend with him about his work. You know, I hope he will get a Nobel Prize about his work. So Rafi discovered the cannabinoids in the 60s, discovered cannabis and the CBD, and then the THC, and then other cannabinoids, and then he was part of the discovery of the endocannabinoid system. So Rafi is, you know, is in the other side of the country of Israel, which is 1 hour drive, so it's not a big problem in Israel. And so I went to Rafi and I started to learn deeper and deeper. And I realized that even though there is knowledge, there is no tools to do the research. So our major problems in the beginning were that you getting plant materials from growers, you harvest them, you're doing extraction, you're taking this extraction, using, and you see phenomenal results. Doesn't matter if we're talking in epilepsy, in Alzheimer, in cancer, okay?
Yeah.
How can you repeat the experiment? How can you get the same plant materials? And what is contained? Every types of cannabis, it's actually kind of a different medicine. Every types of cannabis contain hundreds of different of active compounds. We're changing, degrading. How to keep the cannabis? I'm doing an extract. What is the shelf life of cannabis? To keep it in 4 degrees, -20, room temperature? How long it's going to last? What is the bioavailability? But the major problem was to analyze the compounds, to know what I'm holding in my hand and how it's affecting the cells. And if I'm summarizing 5.5 years, I think our biggest achievement is creating tools to do the research. We started to build a library to look on all the phytocannabinoids from the plant. When we started, we could look around, I think 11, and then it was 17 phytocannabinoids. Today we can see more than 140. So when I'm taking a cannabis extract today, a cannabis flower, and I can look into it and to say, okay, these are all the phytocannabinoids, these are the terpenes, these are the flavonoids. I'm not blind anymore. So it's funny, we kind of played Sudoku. We said, you know, this cannabinoid, this THC looks like that in our equipment. This CBD look like that. This CBG look like that. This CBC look like that. So THCV must look like that. And we looked on that in the mass spec. And then when defined, we said, okay, if THCV look like that, THCVA must look like that. And we looked for that. And for a year and a bit, we played Sudoku and built the whole picture. And when we finished with the phytocannabinoids, we said, okay, now we need to do it for the terpenes and the terpenoids. When we finished, we said, okay, now we need to do it for the flavonoids. And we finished with that. We said, just a moment, the cannabis affecting the endocannabinoid system. I want to see how it's affecting the human body. but I don't have any tools to measure that. I can look on 5, 6, 13 endocannabinoids. How many endocannabinoids is really out there? Okay.
Right.
So again, we started to do the same thing for blood tests, for biopsies, for things from tissues. And we developed method to analyze. Today in a blood test, I can analyze around 150 endocannabinoids, different endocannabinoids. So it's a mess. Okay. And also the receptors. People used to look like there are two receptors for the endocannabinoid, CB1 and CB2. But we know that GPR55 is part of that. We know that GPR 119. We know that the TRPV1 are participate. That okay, I want to look at the whole picture. Let's see which receptors are part of the endocannabinoids action. I don't know how to call them endocannabinoid system, but endocannabinoid action. So today, my lab looking on 38 receptors every time that we are doing changes. So having that kind of a big picture that I can see the phytocannabinoid, I can see terpenoid flavonoids, I know how it's affecting the human body, looking on all of that together. First of all, give me the tools to ask questions. First
Yes.
of all, okay? And then the questions are starting to emerging. Of course, it's everything is in parallel. It's not that I did just chemistry and that I'm still biologist. And then, so having these abilities and these tools, first of all, change the lab in a way that many scientists, physicians, collaborations, companies approach me and said, just want that I'm doing this experiment, but I know nothing about the molecules that I'm using. I can't, I purify them. I can't work with them. I don't know the dose that I'm using. Let's collaborate. And this opened the door for me in the last years to enter to many different angles of different illnesses and diseases using cannabis. The best neurophysician in the world approaching me and said, "Hey, Dedi, I'm doing this experiment. I need you with me." You're not saying, this is this is an
Okay.
honor for me. I'm with you, So more and more, my lab changed in the way that new type of research starting to grow. And there was things that we saw out there, like with the patient, epilepsy or a sleep disorders or multiple sclerosis, that we saw the effect on the patient and said, just what I want to understand now the mechanism, why cannabis affecting which compounds, how to improve that. So my lab grew from a lab of 6-7 students to a lab of 45 students.
Wow.
Which have different groups. I have a group of chemists that doing all the analysis and everything. I have a group that doing cancer biology, and we can talk about it deeper if you want. I have a group that doing research around neurodegenerative diseases like Alzheimer, epilepsy, sleep disorder, multiple sclerosis. And I have a group that's working how cannabis affect the immune system. On part of that, I have few types of small. I don't know if my PhD will think about him small, but that it's not a big group, that it's individuals that doing other things. And in the last 5 years, another big project that we did, we created a big database in Israel on the patient. So in the last 5 years, every cannabis and being picked in Israel, again, there is a problems to be a small country. You need to fight for your borders every day, but there is benefit of that everybody knows everybody and everybody's working together. So until last year, we had just 8 authorized growers, just 8 greenhouses that grow cannabis. So it was easier to work with them, to follow up. So every cannabis that being picked in Israel, every cannabis that every product that the patient can get go through my lab first. We analyze all the phytocannabinoids and the terpenoids, and on the other side, we follow up on the patient how it's affecting them. So does it improve appetite, improve sleep, mainly kind of the side effects, which I don't call it side effect. If you have a problem of sleeping and improving sleeping, it's not your side effect. But we measured that, and we started to bring that together to complete it, to try to match which type of cannabis and which profile of cannabinoids affecting which illnesses and why. And this we took to the lab to animal model to prove that and to understand the mechanism. So we kind of doing a study which is back and forth, which
Yes.
usually scientists stuck in their lab, and then their patient have 4 legs and a tail, and we sacrificed our patient in the end usually.
Yep.
And my lab is working in a different way when there is interaction always with the patients and the physicians. So as a lab, academic lab, I have 4 nurses that I'm paying their salaries in the hospitals, that they are kind of the cannabis phase, or the cannabis nurses in this department. I'm paying the salary, they're part of the lab, and they are sitting, let's say, in the main pain department in Israel or in the oncology department of Rambam Hospital. And they are helping the physician to choose the right cannabis. They are helping the patient to understand better. It's not that we know what to give to the patient.
Yeah.
We are not there yet, but we can help, we can guide. We can say, hey, this probably not the right thing, start low, go slow, and all of these kind of things. You never smoke, try maybe to evaporate, to take oil, to explain, to guide. What I'm getting from that is a relationship, directly relationship with the patient and the physicians, and being with my hands, in the mud.
Yes.
I know all the physicians in Israel that work with the cannabis. I know the patients. I know that, and a lot of things that we feel from the field. That this is helping here and that. We're taking to do a research in the lab,
Yep.
and the other way around. If we discover something unique in the lab, it can be in a week or few weeks already. Patient can start to get it.
Applied.
Today, the 2 main cannabis strains that being given to epilepsy, the 2 main strains in Israel, which I think more than 90% of the epilepsy kids getting them, discovered in the lab. The grower gave me, said, hey, Daddy, I have new breedings and I want to check it. And I came back to him and said, hey, this is amazing. This is helping on the mice, on the epilepsy, like crazy. Look, these are unique compounds. I believe this is what it's doing. And because the patient getting prescription written on the cannabis, it's kind of open door, the broker said, okay, it's high CBD strain, take that. And so there was, I would say in the last 5-6 years, there was few places that we literally changed the way a patient getting cannabis in Israel, what they're getting, doses, regulation, things like that, which is kind of unique for academic lab.
Absolutely, yeah. And I mean, what you're describing excites me for several reasons. I mean, one, my background with cannabis is in laboratories, primarily looking at the analytical chemistry side. So what your lab has done is what I, as an analyst, have dreamed of having the tools to do one day, to actually really dive into the minor constituents of the cannabis plant, to really do in-depth profiling on cannabis products. And one thing that I've seen here in the United States that I view as kind of a lost opportunity is there's not a lot of good collaboration between the analytical labs that are working with cannabis products and trying to profile them or clear them for safe for public safety, or whatever, between that and the hospitals and the academic institutions. They're all disconnected and not really working together, which on one level means that we're not getting a lot of good data that we could be getting. But also, one thing that's always bothered me is just the lack of the the disjointed testing standards between states in the United States, that all of these analytical labs are doing different things, using different methods, usually only focused on maybe up to 10 cannabinoids at most. Most are not testing for flavonoids. And the terpenoid testing methods are often not validated. So your lab is, to me, a really great model to look at. And I hope that here in the United States, I hope that states that have medical cannabis programs or that are legalizing cannabis realize the opportunity that there is to establish a model like this, where you are pushing the analytical testing standards further, that you're trying to marry these data sets together between what's happening in the hospitals, between patients and healthcare providers, and what products they're using, and making sure those are well characterized. So there's an aspect to what you do that I find really, really inspiring on the analytical testing side, and just looking at medical cannabis programs in general. I love that, your situation being in a small country, that you're able to know what's going through the medical cannabis system, what patients are receiving, and then getting that feedback so easily and quickly, and being able to critically think about that, and then develop research projects to examine those mechanisms, then bring those insights back over into the clinical practice and try to apply them. It's very revolutionary, and I hope that people listening to this, especially people that maybe are in some positions of power, regulators and things like that, really see the potential there, because it's really incredible. And I guess one of the things that we'll dive into right now that you mentioned is, specifically your work on cancer, and then eventually I want to loop back around to talk about some of these analytical methods that your team has been responsible for developing. But I guess we'll start kind of broadly and then hone in on details. But from what you've studied, how do cannabinoids and other constituents of the cannabis plant seem to affect cancer cells of different cell types?
So, first of all, it's still a mystery. And I want to, I want to say a few words before I'm starting to dive into details in the cancer research. Cannabis affecting the endocannabinoid system, and the endocannabinoid system is expressed in every cell in our body. I'm not familiar with any cell that's not expressing the endocannabinoid system.
Yep.
As I said, we have more than 100 ligand, more than 100 endocannabinoids, and I don't want to say real numbers, but I would say more than 30 receptors. Okay, so it's extremely, extremely complicated system in our body that balancing almost everything in our body. It's in every cell, and it's balancing how we feel appetite and pain and everything. And then the cannabis is producing hundreds of active compounds that can facilitate that. So if usually we are used to look on, from the 30s of the last century of single compound, single target, something like that. Yeah. This is a multiple illnesses, multiple targets, usually in academia, laboratories, but it's also, in factories and businesses, are very, very specific. My professor from my bachelor used to say that an expert is somebody that know everything about nothing. So usually you're studying one thing and looking a very narrow approach, and it's a very, very difficult to open them and to look from different angles. And first of all, it's a change of a concept.
Yeah.
Second, even technically, it's not easy. You're not an expert for chemistry, cancer, epilepsy, Alzheimer. You can't do all of that, but it's all related because you're affecting the immune system, that affecting the neuron, that affecting the liver that produce insulin. And you need to understand all of that in order to understand the activity of cannabis. And this is one of the big limitations of doing this research. It's so messy that physicians, real companies, not the I don't want to say that cannabis companies are not real, but I'm talking about
Yeah.
the huge pharma and huge companies understand how much it's complicated and how much it's messy. When I'm collecting data on patients, I'm collecting more than 150 products, different products. It's not one medicine. I have 150 that every one of them contain dozens of different active compounds in a combination. And now I'm not looking how it's affecting appetite. I'm looking how it's affecting appetite, sleep, pain, multiple sclerosis, IBD like chronic colitis, autism, PTSD. I'm just starting the list, right?
Right.
So
Yeah.
you can collect 20, 30,000 patients and ending with a group of 100 that got this plan for this kind of illness and that. So it's very, very, very difficult to extract the knowledge from that.
Yes. Yeah.
And it's very difficult to understand how to treat the patient. And there is a lot of unknown, there is a lot of emotions in this field, you know.
Yeah.
And when you going to the cancer, and this is my, entering to this field, when you're going to cancer, first of all, there is tons of emotion around that. The
Yes. Yeah.
Rick Simpson story and stories about patients that being cured by taking cannabis give a huge promise. But how much of this is real is unclear. I know people that started to do yoga and cured from cannabis, from cannabis, cured from cancer. I know people that started to being religious
Yeah. Yeah.
from cancer. I know people. I know personally somebody from my family. She went to the desert. She didn't eat for a whole week, and then she was living near the beach for like a few weeks and drink water from the sea because the salt is cleaning her body. And when she did after the cancer testing, she was kind of a miracle. Does drinking water from the sea curing cancer? The answer is no. There are always a percentage of things, we call it placebo, we call it different names, okay?
Mm-hmm.
That there is success that we don't understand it. The
Yep.
question is the percentage. If 0.5 of the people being cured by taking cannabis, the meaning that every 2,000 people it will help them. But when you're looking on Facebook that you have millions, this number become 50 people, 60 people that are doing a lot of noise. And it sounds amazing,
Mm-hmm.
When you're going to the field, when you're going to the hospitals, when you're going really to the patient, and I'm doing that, believe me, I know that these patients face to face, hundreds and thousands. We don't see success, and this is very sad. Okay, so when I followed in Israel on Rick Simpson oil patient, and I did kind of experiment, not experiment, exam on 150 different people that used Rick Simpson oil. And if the audience don't know what is Rick Simpson oil is a very concentrated cannabis, usually starting with the high amounts, hundreds of grams. You are extract that, ethanol extraction, concentrate that, and taking the amount is quite big. Usually it's high THC, not always. Usually it's sativa, not always. There is no rules for that. And it's usually very, very black and very sticky, so people put it in sarin to take it with their mouth. And there is a guy named Rick Simpson that claimed that he cured himself with cannabis. Probably he did. Very nice guy, I met him. And there is kind of a movement of don't use chemotherapy, don't use the real treatment, go and use cannabis. So when I followed on 150 patients in Israel that got Rick Simpson, I collect the syringe from them and I looked on the results. And first of all, none of them took, the things that they take was totally different. There is no correlation between one syringe to the other one. It's all called cannabis, but totally different medicine. And second, for 150 patients, I didn't see even one success. I didn't see
Wow.
even one improvement. And when I started to look bigger, in Israel, there are a few, I call them partisans, like pirates, that creating this Rick Simpson oil. It's kind of illegal, but when you prescribe under healthcare 500 grams of cannabis for a patient that the government paying for them, I don't know the guy that can smoke 500 grams of cannabis in a month. He making Rick Simpson oil from that, and we all know that, and we approve it. Okay.
Yeah.
So when I started to follow up on a bigger level and in the country level, and I went to this guy that making the Rick Simpson from the patient. There are kind of 5 guys like that. I know they're coming to get help from my labs from here and there. I know them. I'm a friend of them. I started to collect. I came to them and said, "Hey guys, give me names of 5 patients that you really cured. I want to go and to check genetically if they have, I'm a scientist. Let's try to group them and to understand why work on them and that." There is almost none. Okay. Oh no, I had this guy. I said, okay, where is he? No, he died already. What do you mean? So why do you think you cured him if he died? Right? This kind. No, but he lived more. How do you know that he lived more? How did you had the day that he should die before and you know that he lived more? What do you mean he lived more? This is not professional. This is not right. So, and these are not physicians. You don't even know exactly his disease, his situation, how you decide that he lived more. Like it's fit to your profile. So I was very, very negative until now, okay, in this podcast, saying about I don't see first of all, I don't see success in the field, and we need to remember that. And this bring something that is important to put in our mind. When we're reducing pain, when we increase appetite, when we are improving sleep, the patient is taking the cannabis. If it's helping him, good. If it's not helping him, he will find something else. When we're talking about disease like cancer, we are playing with people's life. If we are giving hope on something that is not real, just because we have Facebook and we have that, this is scary and this is not fair. Okay. Yeah. I have people, sometimes I'm coming to my lab and people are waiting out of my door, and I say, hey guys, I'm not a physician. I'm not getting appointment, But sometimes it's a mother with a child, 8 years old, it's coming to me. I have 4 kids. That's it. It's killing me on that spot. Okay. Tell me, look, there is nothing to do. I am hopeless. Please help. Okay. It's not fair. Okay. It's not fair to fool these people. She need to find the best treatment for her daughter. If I think cannabis is the best treatment, I will kill people that won't give it. But I'm not sure yet. Okay. Yeah. So in order to be sure, we need to do a very accurate research and to understand how it's working. We need to understand that cancer, it's a given name to 100 of different diseases. Okay, it's not one disease. If you're taking breast cancer, just breast cancer, you have estrogen plus, progesterone plus, hair plus, triple negative, BRCA mutation. You have, in glioblastoma, there are, what I want to say, every cell in our body will have its own cancer, okay? Yeah. Skin cancer, bladder cancer, prostate cancer, every type of cell. So we already have more than 200 types. And every type has subtypes. So if we are in a breast cancer, you have estrogen plus breast cancer, progesterone plus breast cancer. Every one of them, it's a different profile. We look on that as a different disease. We put it in a cluster. We said it's called the 6 hallmarks. Every time that there is 1, 2, 3, 4, 5, 6, we will call it cancer. But it's not one disease. We're not treating it the same. So first of all, as a concept, you need to understand that cannabis won't solve cancer. Maybe it will solve a type of a cancer or a type of disease, because sometimes they are opposite of each other. Second, we spoke about it already. Cannabis, it's not one molecule, it's not one plant. In my lab, I have over the 900 different types of cannabis, and I have over the 900 and not over than 2,000, because there is a limit how much I want, how much I can screen and work with. Okay, so
Right.
almost
Yeah.
unlimited. So which cannabis can affect which cancer type and how? This is what we're trying to do here. Okay, in a very very deep research and accurate research. So the way we approach it, we're taking a type of a cancer, a subtype of cancer, and we're taking 100 of different cannabis plants, and we first of all do a fast screen for a few weeks, trying to see if there is something unique, if there is one types of cannabis or few types of cannabis that are very potent to this subtype. And when we
Yeah.
see that, we're trying to understand, first of all, if it's a specific action. So I want to say a few things. I'm not against, and I really believe that cannabis can help cancer. I'm the one that gamble his career on that. So believe me that I really hope that I'm not a mistake. But when we are looking for something very specific, every material in the world will kill cells in a plate. If you're taking sugar, if you're taking salt, and you put on all these cells, it will die. I was the head of the botanic gardens and the medicinal gardens. I know many plants that can kill these cells. Okay, so if you're taking other plants that can, have a toxicity or anti-cell, I don't have the right word for that in English. So if you're taking them and you're doing an extract and you put on the cell, there is something we call IC50, how much you need to put in order to kill 50% of the cell.
Yeah.
And every material eventually will kill the cells. The question, whether it's in the therapeutic window, whether
Yes.
if you're taking a rosemary, and I'm always give this example because I did it really with rosemary, you need to put 60 milligram, whatever. If you're doing the calculation, how much a person needs to take a day, it's around 40 kilogram, which is not a therapeutic window. Like nobody can eat 40 kilograms of rosemary. And it probably will kill other things in our body. Okay?
Right. Yeah.
When we use cannabis in the first time, we put 1 microgram of a whole extract, so the molecule was in nanogram, and was very effective, cause apoptosis in the cells to keep the cells like that the cells will commit suicide. And when I calculate the active compound, it was less than the most severe chemotherapy that I have in my lab. So this amazed me. Okay, I said, this is an extremely potent material, which I already know that it's not toxic in people. People are using cannabis in that level.
Right.
Okay. So that was, if you ask me what was my Eureka 5 years ago, it was that experiment that I did still with my hands, which I said, it's crazy how potent it is. Okay.
Yeah.
How much it's not toxic for one hand and how much it's toxic from the other hand. And then, so if I'm coming back to our method, we first screen a lot of cannabis and looking for the most potent to this specific type of cancer. Yeah. And when we define it, we're trying to define why it's specific to this cancer. Because this breast cancer is an estrogen plus, because they have a BRCA mutation. What makes this specific cancer being specific for this cannabis? Okay. When we're defining that, we're starting to go deeper and deeper to understand, from the one hand, the cell biology, the mechanism, why the cannabis is affecting these cells and not other cells. Understanding the mechanism always open a huge gate to treat other illnesses, other cancer, other types. And this is not easy. This is when you have something like that, usually you did this is the big thing. On the other hand, when we define that, we're looking what are the compounds, or what are the combination of compounds that you need in order to do this action. Whether it's one compound from the cannabis, 3, 7, the whole extract. I want to know what are the minimum compounds that doing the maximum effect. Okay.
Yeah.
People used to say that nature is don't touch the nature, it's perfect. Nature is far from being perfect. This is the reason there is evolution. Okay, we're
Yep,
improving, and what is
absolutely.
not perfect is disappearing. I'm always said if we were perfect, we wouldn't have a teeth, we were the beak or something. But like
And there's all
teeth,
sorts of
colors,
examples like that. Yeah, there's
except of kissing, I don't understand this system of teeth and everything. Anyway, so nature is not perfect. Who said that the combination that I have in this plant, which is extremely important, is the best? Maybe if I will change the ratio, I will get better action. So all of these questions we are asking. So I can give one or two examples of a research that we did like that. One of them is in leukemia, which harboring Notch1 mutation. It's a specific mutation in the cell that 50% of the leukemia of the TLL harboring that, but 3% of the glioblastoma, 2% of the breast cancer. So learning that a specific type of cannabis affecting the Notch1 mutation, teaching me, first of all, that I can treat 50% of the leukemia. But in the glioblastoma, if I know their mutation, now know that these have Notch1 mutation, I can treat. So think about it. If you have 2% of people in glioblastoma that harboring Notch1 mutation, if I would treat all the people with the glioblastoma, I will have, in the best thing that I can think, it's 2% success. But if I know to find these ones, it's already become 100%. So this is the reason it's extremely important to understand which mechanism you are attacking. So because you told me in the beginning that most of our audience are people with knowledge in pharmaceuticals or in science, I will go a little bit deeper on that to explain the level that we're trying to. Okay.
Yeah.
So Notch1 is a protein that's sitting on the membrane of the cells. It's a receptors that one of his rules is feeling if there is another cell near him. Okay. It's like it's touching the other cells. I'm flattening here a lot of the science, but
of course, yeah.
Okay. And he's feeling there is a cell behind him. There is something called contact inhibition. When cells have a contact around him, he knows that he can't proliferate, he can't create another cell. One of the things in cancer, you're losing the contact inhibition, and then cell growing on the cell, growing on the cell, create the tumor. Okay. Yeah. So this protein, the Notch1, is one of the proteins that are responsible to feel this contact. If there is a mutation in this protein that is now not sitting in the angle that it needs to sit, it's not feeding the other cells, and the cell think there is no contact. There is another protein that cleavage that. This protein, this Notch1, going to the nucleus as a transcription factor directly, and transcript producing proteins that are important to proliferate the cells. Okay, and this is called the driving mutation. It drives the cells to start to proliferate and create cancer. Okay, one of the protein that is signaling to the cells to create it himself. It's called the feedback loop, because this protein was on the membrane, was cleaved, going to the nucleus, and the nucleus said, hey, produce all these proteins for proliferation, but send another one like me back to the membrane because I'm not there anymore, okay?
And then the whole cycle continues.
The whole cycle continues. This is when you have your mutation. When you don't have mutation, it's sitting on the membrane and feeding the other cells. But if you have a mutation, it's a cycle that's always running. In order to go back to the membrane, it needs to cross the ER and the Golgi. The Golgi is the post office of the cell, okay? The protein is reaching there, getting a stamp, you need to go there, you need to go there, you need to go there, taking a vesicles and go through the microtubules to the right place, okay? So we find out that the cannabis is elevating a protein in the Golgi that blocking, let's say like that, it's blocking the ability of the cells to put the right stem on the Notch1. So the Notch1 can't reach back to the membrane, going to degradation, then you don't have this Notch1 anymore on the membrane, and you're blocking these cycles, and the cells going through apoptosis will start to kill himself. Okay, it's something he already obligate to this circle in this
Right.
mutation and you can't live. So if you're a normal cells, we're not affecting you. But if you have a Notch1 mutation, the cannabis is blocking that on the Golgi, and this is the reason the cells die. Think about it. This is a unique strain that killing cancer, but if you don't have a Notch1 mutation, it just won't affect you at all. This is a CBD strain, not potent. Nobody looked on this strain before, not unique in any parameter, not high CBD, nothing. Okay? But if you are a specific patient with a specific mutation, this can be the cure. Okay. On the other hand, now I have a strain, high CBD strain, with 70 something molecules inside, and I'm asking myself whether there is a unique compound there, a combination of compound, or I really need the entourage effect of everything, flavonoid, terpenoid,
Right.
cannabinoid. So what we're doing, we're starting to do fractions. We're taking this oil. We have a clear phenotype. This killing these cells. Okay. And just to I will finish with this first. So we're taking the oil and we cut it to 4. We said, okay, we have 80 molecules. Let's divide them to 20, 20, 20. Which we have the equipment to do it. It's HPLC preparative, it's PCP, we have all the equipment. So we cut it to 4 pieces, and every pieces we put in different tube. Now I have 20 molecules, 20 molecules, 20 molecules. First of all, as a control, I'm taking all these pieces, put them together, and see that it's back working, that I didn't destroy anything. Okay?
Yeah.
And then I'm starting to do a combination. I'm starting to put every one of them alone. 1 and 2, 1 and 3, 1 and 4, 2 and 3.
Yeah.
I'm doing all the combination, and I see whether there is one fraction that's working or I need a combination. Let's say now I see that tube 1 and tube 3 bring back all the activity. I know that all the molecules in tube 2 and 4, I don't need them anymore. So I already eliminate 50% of the compound. I'm going deeper and deeper and deeper and doing this elimination. Today, I'm doing it in the very efficient way. It's taking me between 2 to 3 months to define the molecule or the combination of the molecule. Doesn't matter if we're talking about cancer, epilepsy, or other things. Okay.
Yeah.
So you narrow it down and you find the molecules. In this specific Notch1, we define 3 cannabinoids. Cannabinoids. 2 of them are known cannabinoids. 1 of them is unknown cannabinoids. We are the first one to analyze, to discover, to send to NMR to understand the structure, okay? But you need the 3 of them. If you put every one of them or every combination of 2 of them, it's not working. You need the 3 of them. And we know already they are binding 2 different receptors. Activate an ER stress that release calcium. We understand the mechanism, but we already understand also why you need more than 1 of them, that you need the 3 of them. Because they're binding different receptors and activating different pathways that create together something in the cells that's doing that. And this is kind of a proof of a concept for me. We didn't need all the plant, but we need this combination. And actually in Israel, I had just 1 plant that had this combination. After I understood this combination, I said, okay, let's take a plant from here that had 1 molecule and a plant from here that had 2 molecules from that, combine them and see that I'm getting the same action. And the answer is yes, I'm getting the same action. Exact. Okay.
Wow. Yeah.
So we took it to the mice model. We did everything in the mice model. We creating the tumors. We did actually 4 different mice models, including a bone marrow implantation to create this leukemia. And it's working perfect. It's killing the cells. It's reducing, it's curing the mice, the leukemia in the mice. And now we're starting clinical trials, literally clinical trials with this strain and this molecule. So this is another question about pharma versus cannabis. Okay.
Yeah.
Now there is 2 roads. I finished everything that I know on the preclinical with it. Okay. I don't have literally almost other things. I'm playing with the ratio, trying to see other things. But I did 4 different mice models for that. I did hundreds of experiments. I understand the mechanism. I understand the compounds. I understand almost everything that I wish to understand as a scientist. I just don't understand why the editor of Cell don't think like me, but that's a different question.
Yeah, that's a really good question.
Yeah. So anyway, now I'm going to clinical trial. In the clinical trial, there is two roads. There is a pharma road. Take this compound, make them purified, doing a clinical trial, phase 1, phase 2, phase 3,
Yeah.
toxicity and everything. 7 years, FDA approved, probably $500 million, or I
Yeah.
will be the richest person in the world, or I will be with zero. It's
Very sad.
very sad. Exactly, both ways. This is one road. The other road is cannabis. Cannabis been given today to around, I'm sorry if I'm not accurate, I think 14,000 cancer patients in Israel as a treatment for improving quality of life. Almost every cancer patient in Israel that suffer from pain, nausea, they will get cannabis or the option to get cannabis.
Yeah.
And I have thousands of patients getting cannabis. So it's already kind of a medicine that's being approved, okay, as a whole extract.
Yeah.
Now I need just to define it to say, hey, this patient, give them this strain, okay? And it's easy, regulatory, it's easy. You're already like in phase 2B, I will call it something like that. You skip everything. You don't need to do toxicity. You can go directly to the patient. You don't need to start with healthy patient and then patient. You're starting with real patient. The patient get on their prescription cannabis. Doesn't matter what you're getting. 30-gram cannabis, 30 ml cannabis. That's what is written on the prescription. Israel is kind of half social. We are not paying from healthcare. Okay? It's been deducted from my salary. I'm paying kind of insurance, but everybody in Israel have insurance. Doesn't matter if you're working or not. If you're not working, probably the people that they working cover that. I don't know how it's working, the tax. But
Sure, yeah.
but when I'm going to the hospital, I'm not paying for that. Okay. When I'm
Gotcha.
going
Okay.
for a physician, I'm paying a simple payment. I'm paying $12. Okay. That you
Wow.
won't go 5 times a day. It's like bags in the supermarket. But but you're paying a fixed price, and we're not paying for healthcare. So when a patient being prescribed for cannabis is prescribed from a medicine under healthcare, you need to understand that. It's not like in US that you're going and pay for that. So we have more than 14,000 patients that getting cannabis under prescription, under healthcare, but the prescription is not defined which cannabis to take. So when I'm going to a clinical trial today, and I'm working in all the oncology department in Israel, I'm doing today 13 or 14 clinical trials, most of them on pain and sleep and foot cancer in that way. But I'm working in this department. I just need to speak with the physician, Gil Barcela, my friend, the head of the oncology department in Carmen Hospital, said, hey, Gil, I think that the leukemia patient with Notch 1, that you don't have any treatment for them today, this strain can kill the cells. He literally, it doesn't sound good, but he literally don't care. Okay, instead of give them Scooby-Doo or Mickey Mouse or White Widow or Deep Purple or whatever names that you call it, I will give them the strain that you. I don't care. It's a CBD strain. It's even not psychoactive. I will give them whatever you want. This is cannabis route. So you can do a double-blind, placebo, real clinical trial, but get permission in Israel between 3 to 5 months starting a trial and see in the real world, not on mice, if the cannabis is really helping this patient with the nodule. Okay? You can be with a product, with a medicine that can cure people in a year and a half on the shelf after clinical trial, double blind, placebo, real trial.
Yeah.
Real measurements, blood measurements, endocannabinoids, toxicity with everything, but without going the Via de la Rosa path of the FDA. And it doesn't need to say that if you're going that route, you can't go in parallel in that route. Okay?
Right. Yeah.
But I'm telling you literally as a person, as a human being, thinking that maybe I have something, a medicine in my hand that can cure now a child with a TLA leukemia, but now I need to wait 7 years,
Yeah.
which you can get it in the backyard, which it can get it in the day, which he just can buy that because he can get it. But I'm waiting this. This is something kind of insane for me.
Yeah.
So I gave you now an example of a complicated action of P molecules. I have a results in my lab on a breast cancer, specific type of breast cancer, but I won't go to the details. I just said that in that way that we narrow down, in the end, we discover a specific phytocannabinoid, that it's one phytocannabinoid that action. But this phytocannabinoid is extremely unique one. It's a phytocannabinoid with oxygen in the in the in the ring. I don't.
Oh, okay. Yeah.
Okay.
Yeah.
So
Sort of in the periphery of the aromatic rings.
Exactly. So you have oxygen inside, so it's probably less lipophilic, more than.
Right.
But this is extremely. It's a new cannabinoid. It's not from the families that the cannabinoid, phytocannabinoid that we know. When we got the first NMR, when we understand the structure of that, we thought it might be a mistake, and we send it again.
Sure. Yeah.
So I think that sometimes it can be one molecule, sometimes it would be 3 molecules, sometimes it would be more than that.
Yeah.
But the ability to narrow down, discover that, open the door to understand, bigger picture and what we can treat. And I think that the cannabis field in general will go in 2 different roads. Okay, if I think about the medicinal cannabis in 5 years, I think there will be specific illnesses that will be treated with a very accurate way, very specific strain of very specific molecules, very specific product. And the majority will still use cannabis as a whole plant. And I will explain myself, okay? If I'm looking on the patient, at least in Israel, but also in the other world, in all of the world, most of the patients using cannabis as a improving quality of life.
Yeah.
Pain patient, PTSD patient, autistic kids, cancer patient that's suffering from neuropathic pain and from nausea. They're all using cannabis for improving quality of life. Just a small portion of patients taking cannabis for a specific indication. A kid with epilepsy that taking cannabis, the majority is to reduce the seizures. This is very specific, but most of the patients taking it for quality of life. Cannabis affecting many pathways in our body. It has affecting many cells.
Yeah.
So when you're taking a PTSD patient, post-trauma disorder, he taking cannabis for reducing anxiety, for improving sleep, okay, and for reducing his nightmares. It's affecting the dreams. It's affecting us to get into the REM, the dream sleep person. Okay. If we're looking about a cancer patient that getting now chemotherapy, is getting cannabis to reduce nausea, to improve sleep, to reduce pain, to improve his mood, to improve his appetite. It's not one thing. It's not one compound or 3 compounds for a single thing. It's a little bit improving his sleep, a little bit improving his appetite, a little bit improving his mood, a little bit reducing his pain, and all together, you get a big action of improving quality of life. And I don't think that we will be able to define the specific molecules because it's not specific molecule. These 2 molecules binding these neurons and then you're sleeping better. These 2 molecules are binding these receptors and helping in improving appetite. And it's a complex of things and on that I believe cannabis will stay as a cannabis, as a whole product. maybe the package will be beautiful, with the Star
Sure.
Wars
Yeah.
pictures on them and other things. And maybe we will find a better delivery and the vipers will
Mhm.
be better and this. But in the end, we will stay probably with the whole extract. On the other hand, a cancer patient, breast cancer with estrogen plus profile might get a single compound or a combinations of compounds or a strain that we know exactly which compounds inside and what is the portion of the compound
Mhm.
as a treatment to this cancer. Why? Because now we less care about her appetite, that we need to save her life. And this is very
Yes.
accurate measurement. And I believe that in 5 years, we will see on the shelves these two products more and more accurate, knowing exactly the
Yep.
compounds and everything, and things that are more general, or
Absolutely. Yeah, I mean, you're highlighting something that I've speculated on. You're basically nailing it on the head. What I've mentioned before on the podcast, that I as well see room for these highly standardized, unique formulations targeted for very specific uses, generally for acute disease states, very
yeah,
acute issues. And then herbal cannabis has its place exactly in what you're saying, in this sort of more chronic quality of life, multi variate, situation that is always going to have this level of complexity to it that is essentially impossible to tease out anyway. And I mean, there's so much that you just covered that I want to come back around to. I mean, one question that I have that I think is on a lot of people's minds are what what what's the role for the quote unquote minor constituents of cannabis, and I guess I'll say non-cannabinoids, because we've mostly talked about cannabinoids, but are you seeing roles that terpenoids, flavonoids, and even going beyond those, sterols and stilbenoids, these other non-cannabinoid compounds, do you see them playing any role in some of these activities on these different cell types that you're investigating?
yeah. So I will divide my answer first of all to the cannabinoids, even though you asked not
Sure.
about
Yeah.
them. Minor cannabinoids is terminology that I don't like,
Sure. Yeah.
because because if I have in a strain a cannabinoid name, I don't know, CBGM or
Mm-hmm.
CBGO, or a cannabinoid that you never heard about that name, and we call it 32918.
Right.
Okay, if it's in a specific strain, if it's 2.5%,
Mm-hmm.
it's not minor. It's the second cannabinoids expressed. You never heard about it. It doesn't mean that it's minor.
Right.
And 2% in a plant, in a dry flowers, if somebody is using 1 gram, which is the average of a joint, it's
Mm-hmm.
0.75 1 gram. If a patient, the average in Israel for patient is 32.4 gram a month. So it's around 1 gram. Okay, so
Yeah.
I'm always calculating that way. So if a patient taking 1 gram that have 2% of a cannabinoid is getting 20 milligram a day, which is not
Right.
minor. Okay,
Yeah.
it's already not minor. It's quite a lot. Okay, so the definition of minor or not, we call what we are used to say THC, CBD, CBG, CBC, CBN. They are the major. But the
Right.
minor is because I don't know how to look at it in the HPLC. I will call it minor. So first of all, I have a problem with this definition. Okay, and we see in many studies that these, what we call just minor, are extremely important,
Mm-hmm.
and they are major and not minor, and sometimes they are extremely potent, more than THC, CBD, and other cannabinoids. So first
Mm-hmm.
of all, on phytocannabinoids, I think they are extremely important. Okay, when we're talking about terpenoids and flavonoids, it's a big question. In just one study that we did, we find canaflavin A as important.
Mm.
And so again, let's divide this answer into terpenoids and flavonoids, okay?
Absolutely.
Flavonoids, there are unique flavonoids in cannabis that we don't find in any other plant.
Yeah.
What makes them unique is the cannabinoids because it's become unique. And part of them, we know that they are important and potent and doing action. I'm almost not familiar good research that being published that they are important to a kind of an illness or kind of you know the reasons that people using cannabis that the flavonoid is part of that. I don't know, appetite, sleep, pain,
Sure.
whatever. We find it that it's affecting microglia in the brain and these kind of things, but it's a study that we're still on. So except that, we didn't find the other flavonoids. We are looking around 40 different flavonoids. We didn't find that they are important in action. It doesn't mean that they are not. Just say that I didn't find yet, you know.
Absolutely. Yeah.
The terpenoids is the terpenoids are a different question. And first of all, they are changing all the time in the plant.
Yes.
So it's important when you're picking the plant, how you drying it, how you curing it, how much it's fresh and not fresh. They are changing all the time. So if you're taking the phytocannabinoids, we used to say that they are changing, and if you grow it in different light, different soil, you will change. But what I learned that the ratio between them are almost always the same. So if there is 10-1 CBD to THC, it will stay 10-1. Maybe it will be 10 THC, 1 CBD, and it can be maybe 15 THC, 1.5 CBD. But the ratio is always the same. So, and I don't know how much you hold in your lung, you know, when you take a breath, and so, but you're getting the same ratio, the same complex of phytocannabinoids almost always. And when you're doing curing, you dry it, it's quite stable. Okay. Actually, I can take a seedling, you know, for the flower, just the fresh leaf, and tell you from the seedlings what will be the ratio between all the phytocannabinoids on that stage. So the meaning that it's quite stable.
That's what I used to do for farmers in Oregon.
Okay. But when we're talking about the terpenoids, it's a totally different story. I don't have a clue what you will get when you pick it. If you pick it in the beginning of flowering or the end of flowering, it will be totally different. On you when you're doing when you're curing that, it's totally changing. And after 1 month, a little bit than 1 month, there is a reduction of almost 90% of the terpenoid. This is a paper that we just published in Tips of Plant Biology that are, we checked for 2 years the stability of the molecules, the terpenoids and the cannabinoids, whether you put them in a shelf life, -20, -80, -4, you know, oil, olive oil versus canola oil versus, we did many combination and look how the compounds is changing, what is the degradation, what is the shelf life of cannabis, how is the best to store it, you know, all of these questions. Ground flowers versus whole flower. I have a student that did many of these actions in order to check that. The terpenoids are, you lost almost 90% of them after 1 month. So the question is how many of these terpenoids really the patient is getting.
Yeah.
Okay. How much when you're smoking a joint, when you vaporate, when you keeping the cannabis, and usually the patient, at least today in Israel, will get the cannabis around 4 or 5 months after it's been picked because it's need to go through a process and regulation and the GMP and everything. It's not like we had 3-4 years ago that it was from the field to the patient. Okay. The way they curing it and everything, a lot of these different. So first of all, the question is how much is really left there. And when you're doing extraction, whether it's ethanol extraction, butane extraction, you lose many these terms.
Yes. Yeah.
So altogether, it's a question how much really the patient are feeling the terpenes. You know, we know that you feel it's the smell, it's the taste, of course there is. And it's for me, it's a little bit disappointing. I'm working on that a lot. I invest a lot of time and money, and I'm still in none of our studies, another matter which type of study, I didn't put my finger on terpenoid that is part of the effect that I see. Again, doesn't mean that it's not. It means that I didn't put a finger. I'm far from being, uh, that's what I didn't succeed to see that. This is the result. Now, if you ask me if I think they are important, the answer is probably yes. People call sativa in indica. Okay, what exactly is sativa in indica? As a as a plant biologist that did genetics and in plants, I don't know to define that sativa in indica. It's 70% indica, 30% sativa.
Oh gosh.
What the meaning?
What does that mean?
What does that mean? How do you describe that? What it's they have the root is sativa and the leaves are indica. What the hell?
Yeah.
You know?
Absolutely. Yeah. It's something that's, my background initially being in botany, that's something that I've had a hard time wrapping my head around. And I've seen some of the good work that, like John McPartland and Ernest Small just put out a paper to try to clarify the taxonomy versus the vernacular of those terms and to try to find some standardization. But it is extraordinarily confusing. And especially if you're trying to parse through old research and particularly taxonomical and botanical research around cannabis, and you see that through the past 100 years, different people have used these terms inconsistently. You don't know exactly what they're talking about. And even if you go back, even further, you have different taxonomists that have classified cannabis in different ways, sort of operating in isolation from one another. So yeah, just the use of those terms is a huge mess.
Yeah, but what I want to to get to is that people define sativa and indica also by the effect. He said,
Yes.
okay,
Yeah.
sativa is lifting and indica make you feel more sleepy and heavy.
Right.
We're joking. We said indica in the couch when you're smoking it, you're stuck
Yeah.
in the couch. Okay, so when you're trying, and this is a work by Arno from Netherlands, when he
Yeah.
started tried to define indica and sativa, and he went to the to the shops, to the coffee shops in Netherlands, and said, okay, give me the best indica that you have and the best sativa, the most strongest indica that you have and the most strongest sativa. And it took, I think, from 50 different coffee shops, 100 strains, and tried to define that. He didn't do that in the level that I will wish to do that, and we can do that today in the lab, because he didn't look on all the variety, but he did what he can. So he checked the phytocannabinoid and the terpenoid and saw that the different are just in the terpenes. The phytocannabinoid looks the same between the sativa and indica statistically, but the change is the terpenes. And what you learn from what we can learn from that the effect of feeling uplifting or sleepy is more affected by the terpenes and not the cannabinoid, or the combination with this group of terpenes with these cannabinoids, or that group of terpenes with that cannabinoid.
Yeah.
So this is teaching that they are important. I couldn't define it yet. I couldn't find. Working with companies, there is a very good company in Israel called Ibna that they are doing profiles of terpenes, and I'm working with them a lot and trying to find, things, but not yet.
Yeah, and I did a really nice interview fairly recently with Vincenzo DiMarzo, and we were talking about the entourage effects and the sort of, I guess, misunderstanding in the broader kind of popular culture within the cannabis industry about, what he mentioned is that when he and Rafi first presented this concept of the entourage effects, they were thinking about 2AG and these congeners that they didn't understand their effects and everything. And And then over time, that concept got sort of reinterpreted to apply to the cannabis plant, and these effects that might be at play between terpenes and cannabinoids and everything. But what Tim and I were talking about now, I've also talked to Arno as well about this, that there's just not a lot of data that actually shows these sort of quote unquote entourage effects within the cannabis plant. That doesn't mean they're not there, and everyone I've talked to makes a point to say that doesn't mean they're not there, but there's very little data that has actually shown the mechanisms of those sort of synergistic effects so far, which is important for people to understand.
Yeah, so first of all, this is true. It's funny that you mentioned that DiMarzo said that, but this is something that is clear. The paper about the entourage effect from 1998 of Rafi and DiMarzo and Professor Ben Shabbat, we're talking about endocannabinoids, two endocannabinoids in the brain, nothing about cannabis. People like, it's not people, I think Ethan Russo published that in 2000 and something, that actually about the entourage effect of the cannabis. And again, Ethan is not another guy from the street. He
Right.
published a lot of good papers about that and showed that from our research, we see the entourage effect. I gave you an examples of three phytocannabinoids, and I know the mechanism and why it's doing that. And I think for me, this paper that we are trying to publish now on the leukemia, it's a breakthrough because it's one of the first times that we are proving the entourage effect and showing that you need more than one compound, that one compound won't work. You need the combination, and explaining exactly why this is binding this is binding that, this is doing that. And this is why I thought it's a very important work. But it's not clear how much this entourage effect is there and in how many cases it's true. We talk about it, about the overall action of cannabis, which a patient taking that and getting many benefits. We need to understand the endocannabinoid system in order to understand the action of the cannabis. The endocannabinoid system, it's a homeostatic regulatory system in our body in most of the cases, not in all of the cases, but in most of the cases. It's balancing things and fine-tune action, okay? If we touch a hot glass and our hand is jumping, the reason of that is because there are sensors in the tips of our fingers that feeling the hot and the pain, sending a signal fast to our brain, that send a signal back to our muscles, okay, to release calcium, to contract, okay?
Yes, yeah.
And they contract. This is a very fast action, electric action.
Yeah.
The cells contract, okay? But from that moment, you don't need this pain anymore. And the body is releasing the pain. It's doing it in a very specific way that cells near these sensors, creating a specific endocannabinoid that bind it and reducing the pain. But this is very specific, very fine-tuned. It's by localization. It's there. Okay, if it was here, it was just here. If it was here, it was just here. Okay, it's very localized, very fast. The endocannabinoids are lipophilic molecules sitting on the membrane, being cleaved, released, binding, half-life of few minutes. Okay, dead, boom, gone, and they are gone. Okay, they're doing their action, they're gone. This is a very specific, very small, but doing the action.
Yeah.
The body have also the severe action. If you now wounded, the hand is totally cut. The body is not playing with endocannabinoids. It's producing opioids, like morphine, right? It's producing opioids. There is a penalty of this opioid. The body now is in the totally stress. You need to save your life, to reduce your heart beating, that you want to lose a lot of blood, that you want to move. You become totally stoned. Maybe you will faint. okay, from this boost of hormones, from boost of not hormones, boost of opioids, okay, you will be totally stoned. Maybe you won't feel the pain at all, okay, but there is a penalty for that because you're totally out, okay.
Right.
So the body have the mild, small, specific action for mild, small things, and the severe action when it needs severe actions, okay. Think about the hunter that ran after the deer 10,000 years ago, okay, in the forest, bare feet. Every rock that he hitting, everything will stone him. The wolf and the tiger will take him quite fast. Oh, I'm out. Probably the war feel like that. They just not
right. Yeah, they got selected out.
So, on the other hand, he's focusing on and the body is just managed that in a specific localization. Okay, I'm always giving the appetite example because I think it's a great example.
Yeah.
If we are not eating for 2 days, for 1 day, our intestine is shrinking and sending a severe signal to our brain hormones to eat. We become angry. We become hungry. We can't think. Okay, why the body is doing that? Because it's in a very catastrophic situation. Okay, we need fuel. This car is not driving without fuel. We call it glucose. We need to burn glucose in order to create ATP that we can move, we can speak, we can move, we can breathe. Without fuel, the engine will stop. Okay, so the body needs fuel. If you didn't eat, it doesn't have fuel. He needs to start to create fuel from his stocks. Okay, unfortunately, he's not going to the fat first. He's going to the muscles, starting to break muscles. Okay.
Yep.
This is a catastrophic action for the body. It's called cachexia. When we're starting to break our muscles. Okay. So if you're not eating for a long time, the body now needs to save itself. And it create a very, very strong, severe action that you will focus on eating. Let's go back to the hunter. He's running after the deer. He's so hungry that he don't feel pain. He don't care about anything.
Right. Yeah.
He need to eat. He's totally focused. Okay, nothing but hunting this deer. Okay, but now there is another system in our body that create appetite, the endocannabinoid system. This is not a severe one. When we walk near a barbecue and we smell the barbecue, so small molecules from the burnt meat entering to our nose, going to our olfactory system, signaling to a neuron that producing specific endocannabinoids that going to our pancreas, that creating insulin, and there is a feedback loop that increases appetite. But this is a mild, If my wife is standing near me and said, "You don't need it now," it's falling immediately, I not need to eat that. If I will move from there and won't smell it after 2-3 minutes, the appetite will go down. Yeah. It's not I'm not eating for 2 days. If I'm going to the other room after I did eat it 2 days, I will still think about food.
Right.
But our dessert is coming when we are totally full, and the dessert is coming and we're eating that. This is the endocannabinoid system. But sometimes we can control, and I'm looking on my body and said, okay, that's enough for me. I won't eat more, even though my body is signaling. We control it.
Yeah.
Let's go back to the hunter. Okay, he ran after the deer. He's so hungry, so that he's hunting the deer. Now he's coming with all the tribe around him, burning this deer, and they're starting to eat. After a few bites, after not eating for 2 days, after a few bites, he will feel totally full. But evolutionary, the body creates this system of creating appetite. Keep eating. You don't know when you will have the next deer. Keep eating. You don't know where will be the next meal. It's important. This appetite is extremely important. Okay, but it's under control because the wolf and the tiger also have an endocannabinoid system. They're smelling the smell. They will come. But when he will hear the noise in the bush, he's totally under control. It's not a severe action. It's the mild action being controlled. He will move. He will do it. Okay. I already
Mhm.
ate. This is a tiger. Maybe I will fight him next time. If he wouldn't eat for 3 days and he will hunt the deer and the tiger will come, he might fight with the tiger. He might lose.
Right.
Okay.
Yeah.
He probably will
Yeah.
lose. Okay. So this is system that the endocannabinoids built to fine tune, to
Yes.
do
Yeah.
very localized, very specific, very fast. Cannabis is affecting that. For that reason, in a lot of cases, it's mild, it,
Mhm.
it's small, it's fast, okay? Because this is the endocannabinoid system, we can't change it.
Right.
But it's many things in our body which are small pieces. So
Yes.
when we're taking cannabis, it's a little bit affecting the appetite, a little bit affecting the pain, a little bit affecting our sleep, a little, and you're getting something that is the all pieces bringing something that is big. And this is, I think, the unique thing about cannabis,
Yeah.
and why in some cases it's the perfect treatment. Instead of taking 6 different pills, take
Yep.
cannabis. Okay. In other cases, we don't need to fight it. It won't be the right treatment. It's not strong
Yeah.
enough. It's not potent enough. It's not. And we need to understand in the end, we want to help our community and the sick people, the people that suffer. That is the bottom line, you know. Many people want to
Yes.
make a lot of money, and I understand that everything is fine. But in the bottom line, if we're going to the, deep in the bottom,
Yep.
we want to help people to live better. Okay,
Right.
and cannabis can bring that in many aspects. I want to say that we are talking about cannabis, but I think cannabis opened the door for medicinal plant in general.
Yes. Yes. Absolutely. I
So go on.
I was just going to say, I totally agree. It sounds like you're about to touch on one of the things that I'm most passionate about, because where my personal work is steering now is looking at other plants, rethinking the term cannabinoid altogether. Like, what does that mean? What is a cannabinoid? What molecules fall into that definition that maybe we've studied before but not thought of in this context? So not to interrupt you, but you're touching on something I'm extremely passionate about, so please continue.
So I think, I'm coming from this world, from medicinal plant. I was the
Yeah.
head of the biggest greenhouse in Israel for medicinal plant for years. And this was my passion always. And I think there is still a lot to discover that.
Yes.
For thousands of years, people use plant as a medicine. There was
Yep.
almost nothing else. Okay.
Right.
They learned that if you're giving this dose, it's killing you. Oh, we lost grandpa. It didn't work. And they lost and they learn which dose is helping you. There is a lot of placebo in that,
Yeah.
but it's also a huge amount of knowledge. From the beginning of the last century, from the 20s, 30s, 40s, when microscope were discovered, then the
Yeah.
equipment become better, people learn about bacteria and viruses and DNA and everything. And people started to bring medicines in a better and accurate way, which is very positive. I'm not an
Hmm.
herbalist that thinking that medicines are not good. The reason we're reaching 90 and 95, I spoke with Rafi Beshulov that was 90 last week. I wish I will be a half from his abilities in that age. And one of the reasons is the healthcare become much, much, much better. We can't
Yeah.
argue with that. But companies and scientists and all the science try to be very, very accurate, very specific.
Mhm.
So they had to simplify everything, and they started from the beginning, from scratch. Okay, let's take single compounds to single target. Let's understand one and one. This is simple. That's what I know to do. More than that, it's too complicated. Okay,
Yeah.
and it worked for many, many, many illnesses and diseases and a lot of medicine. When I have a migraine, I'm suffering every 1-2 months from a migraine. I'm taking one pill of Elvin gel and it's done. So it's working, let's face it. Okay. First of all, people don't know that I think more than 80% of the medicines today coming from plants.
Yeah.
People are telling me, Daddy, why to use chemotherapy? Why not to use molecules from plants? And I'm looking at what do you think, where the chemotherapies come from?
What's the difference?
Taxol is from Taxus and vincristine is from Vinca, and like they're coming from plants. They purified it and making a patent and selling it as a pill, but it's coming from plants, not different.
Yep.
Okay? So, people knew for thousands of years that if you're taking willing widow and extract it, boiling that and drinking the tea, is reduce fever and help you reducing pain. So in the 50s, 60s, 70s, I don't remember the year it's been happened. They took the willing widow, tea, and they defined it that the molecule that doing that called salicylic acid. They're doing few modifications that call it aspirin, and we take it as a medicine today. But it's coming from there. But this is kind of simplified it. Take one molecule, single target, understand the mechanism, make it right. There are few illnesses, few diseases that we are hitting our head in the wall for years and not succeeding to break the wall. It's cancer, it's Alzheimer, it's ALS. I can give more and more examples, I think thousands of laboratories working on cancer research.
Yep.
The discoveries and the things that we succeed to do is extremely minor. Thousands of laboratories, scientists, companies working on trying to cure Alzheimer or blocking Alzheimer. I think it's the most it's the disease that burn the most, the biggest chunk of money that I can think of without any result.
Yeah, that's definitely how it seems.
And it's unbelievable. We don't succeed to do that. And maybe we need to start to think for the different level, not a single molecule, single target, few molecules, few targets, more complicated. It's a polypharmacology. It's already kind of a concept that's starting to emerge. And the best example that I have for that, it's the HIV. That for years they're trying to treat this buster virus,
Yep.
We now know what viruses can do. Okay?
Yeah.
I had a religious professor in my bachelor that teach virology, and he entered with his, the Jewish have this
the yarmulke,
the yarmulke on the head. He came with his yarmulke, arranged it on the head, and said, viruses are like God. That's the way he started the course. That viruses are like God. They are everywhere. They can do everything, and we know nothing about them.
That's great. Wow.
Okay. So for years, we're trying to treat HIV without success. Many laboratories work on that, but there was few discoveries. Now let's block the virus enter to the T cells with this system, and they create a medicine for that. But it didn't work very well. It's improving 20%, the action there. Okay, so let's try to block the virus to kind of divide or proliferate or something like that. And they create a medicine, this is working in 10% efficiently. Yeah. And another, everyone tried to attack the virus from a different angle. Till one day somebody came and said, hey, this is working 10%, this is working 20%, this is working 10%, this is working 5%. This is attacking the virus from there, this is blocking from there. Let's make a cocktail on all of these drugs and give them together, okay? And the cocktail today, it's what is working why these diseases coming, it's not like epidemic as it was, okay?
Yeah, it's been incredible.
So this is kind of a concept, let's not give one, let's give few that giving together. Usually cancer patients today not getting one type of chemotherapy, they're not getting one pill, they're getting a cocktail, they're getting a combination of chemotherapies. And this is the concept. The concept said, let's give few molecules that doing few actions together, okay? Attacking the cells or attacking the system from different angles, make it more complicated for them to defend, something like that.
Yes.
Okay?
Yeah.
Plants harboring this complexity in-house, with them,
Yes.
within.
Yep.
Cannabis is a good example because it's one plant that harboring hundreds of different compounds. But we have many plants that we know for years that people are using as medicines, but because of the complexity, we're not touching them. We're not really using them as a medicine. Okay. More than that, now I'm saying plants that have few compounds that are complex. I can take it to the next level and said when you're going to the Bushman, or you're going to the physician 3,000 years ago, and you had, a stomach ache or you had a fever, he wouldn't give you one plant. Usually, we'll take a leaf from here, a flower from here, a root from here, smash them together and give you. It's true that part of that is placebo, but part of that it's a lot of knowledge of knowing that this is working. And why it's working? Probably because there are few compounds from this flower, few compounds from this leaf, few compounds from this root that all together doing something that is bigger than the one that doing alone. And I believe that we will see more and more and more studies, more and more pharma open the door for that. And cannabis open the door for that in the way that it's a plant, it's harboring a lot of molecule, and it's bring within tons of money. And this is always, the door, my salary is always the same. And I'm even embarrassed to say that here, but the amount of the salary, but the amount that I raised to the lab in the last 4 years give me the ability to recruit a huge team, to buy the best equipment that I can take. And today, it doesn't matter which plant I'm taking. I have a PhD student that collect 96 different algae from the Mediterranean Sea. We did an extract to all of them. We screened them on different diseases. We purified compound from them. For me today, it doesn't matter which plant it is. It can be cannabis, it can be ayahuasca, it can be ibogaine, it can be a psychoactive mushrooms. I have the methodology, I have the team, I have all the equipment, the tools to take it, to analyze all the compounds, to screen on many different diseases or illnesses, to see where it's affecting and started to dive into and understand the mechanism and to do this combination even. Okay?
Yeah.
Part of what I did in the last years, I opened a private company. So I have my own lab that 40-something people, and I have a company that's sitting not far from here that having another 45 scientists. Okay.
Oh, wow. Okay.
So altogether, we're a team around 90 people that's working. Okay.
Yeah.
And the reason I opened the company that I didn't have more space here, and also I need very senior people that will do things that are beyond my ability. When you're taking students, you need to teach them, you need to be to guide them, but to take very, very, very senior people, that can manage a whole research, that I needed it to be out of the academia. So together with the Technion is partnered with this company. We opened Cannasol, cannabis solution or cannabis soul,
Okay.
soul.
Yeah.
Okay. And these companies, first of all, give me a lot of service. I'm buying service from them. If I need to purify the compounds, I want to take cannabinoids and put a fluorescence tag that I can follow up in mice, whatever they are doing for me. But also, we did few things in Cannasol that are trying to open the gate and to open the idea that we just said now, okay? Because in the lab in academia, I'm kind of limited, but in company, you can do whatever you want without limitation. So what we did, one of the things we opened kind of a sub company called MyPlan. What this company did, we create different 20 different cell models for different diseases. Okay.
Mm-hmm.
And we kind of giving a service. You are a breeder today, and you're coming to me and say, Daddy, look, I'm breed amazing. You don't believe what I have. I have a strain that you must test them. And I have it in every conference, at least 15 people like that come approach.
Oh, yes, I'm sure.
What do you mean? What do you mean amazing strain? It's good for make me sleepy or make me alert? You know, it's will. What do you mean? What is good? If I have a high sugar in my blood or low sugar, it's the opposite. What do you mean good? Good for what? Oh no, Daddy, that I need you to check. You know. Okay. So what we did, we developed 20 cell models for different diseases, and I'm giving service. And you have a new breed, you think it's important, you want to test this new mushroom, psilocybin mushroom or whatever, you want to take that, I will run them on 20 different diseases and will give you a report saying how it affects each disease and how it's comparing to everything that I test today. Do you have a blockbuster? Do you have something that we never saw?
Yeah.
Or, you know, it's another thing. It's the average of how it's affecting them. In the COVID-19 now, we had a lot of work because part of our systems is virus infection or inflammation, cytokine storm. So many companies send us materials, not even plant materials, things that they want to test. Okay. So as a concept, I'm giving you this file and say, this is yours. You know, I'm taking $10,000 for 20 sprints like that, 3 repeats, and I give you a file. If you work in a laboratory, you know that I'm losing money if I'm taking $10,000 for 20 sprint, 3 repeats. Okay. But it's fine. Okay. That's what it's yours. You're getting the file. What I'm getting that? What is the business model behind it, or the scientist model? First of all, I'm the first one to see the blockbusters. Okay. To see the amazing things. And usually I will come to the person that sent it to me. He's a grower, he's a breeder, usually is not a scientist, not a professor in the university, and said, hey, John, you don't believe, I never saw a material that blocking insulin like that. This is crazy. It's working 100 times better than the best drug that I saw. First of all, let's try to define which molecules are inside. I'm an expert for that. I can help you, and then you can write an IP, whatever. Second, let's take it to a mice model. Let's take it to real science. Let's do. I'm not forcing you to do that with me. I'm just offering, and I believe that a lot of people will stay and do that for me, with me. So it's kind of exciting to see that. But more than that, and much more important, I'm creating a huge database on many materials, on 20 different diseases, the molecules, and how maybe I can combine them. If we go to the HIV, if I see that this kind of ayahuasca that you send me, it's reducing 20% of the insulin absorption, you know, because it's blocking that. And I will see that this flower is doing that, and I will see that doing that. I can combine it to something that giving a whole as a better treatment. We will never define it if it won't be that way. And it's kind of taking the you know, the old physician or the herbal medicine, you know, the all these tribe physician, and do it the shaman, exactly, the shamans, and do it what they are doing in an accurate, scientific, modern way. But the concept is quite the same. You know, which leaf, which flower, which I need to combine to get a treatment that none of the other single one will succeed. So this is, I totally believe in that. I totally believe in this, you know, word, and it's extremely excited. You know, I started to work in the last year with you know, what people call the magic mushrooms, or you know, psychoactive mushrooms. And again, you know, I never tried it. my life is good enough that I don't want to. I'm kind of afraid or whatever. It's not for me.
Don't want to risk disrupting a good
I
thing.
don't I don't want to risk my brain exactly, you know. But I think, if somebody is taking 2 grams of something and he see flowers, he think he's Avatar, or he think he's flying, it's probably very potent. Okay.
Yeah, to say the least.
And there is hundreds and thousands of these kind of mushrooms. So it's
Yep.
kind of like the
It's very
cannabis
similar. Yeah.
6 years ago, everybody said THC, CBD, cannabis. Okay, guys, there is all the rest, and there is the entourage effect, and there is tons of things. So people said magic mushrooms also been fine, guys. There are hundreds of species, and every one of them doing different things. We understand that but for me, I have all the tools today to run very fast with them. So what I want to say, I had a podcast in Australia 2 weeks ago, and it was more than 2 weeks. And somebody asked me, the audience, that do I think that these mushrooms will be as big as cannabis?
Mm-hmm.
And I told them, guys, cannabis is a plant, mushroom it's a kingdom.
Yes.
We are
Yes.
not. So what I want, my message here, we are not even starting to scratch the beginning
Yes.
of understanding the unknown in the plant medicinal kingdoms, that it was too complicated till now to touch it. And I think
Yes.
we reached the point that we started to open this, this box.
Yeah, I mean, you're describing something that I'm part of my role as an educator is to try to get people to see this and be excited about. But what you're highlighting is that we're in a very special period of time where just like in years before, when new technologies, new methods come about that all of a sudden enable us to look at biology and things in a different way and gain new insights, we're going through sort of a kind of gestalt shift again right now, with these that that you've been such an integral part of developing these new tools, new methods, ways of making these methods very efficient and fast, and gaining perspective. I agree with what you said, that cannabis kind of it opened the door and it gave us a perspective that now can be applied to everything, to natural products broadly. And it's something that when I talk to younger people that are going into grad school right now or about to come out of grad school and are thinking about getting into either cannabis research, or they want to get into psychedelics or just natural products in general, I try to impress upon them that there's so much research to be done that, like, sometimes these students feel like most of this stuff has been tapped. We know so much.
We missed the train.
Right. Yeah. And it's like, well, no, that's a perceptual problem, because when you wrap your head around where we've come from and where we're at now, you start to see this immense landscape ahead of you that is largely unexplored. So much of this research has highlighted our own ignorance and just inability to see. Like I said, when you don't have the tools, you just can't go there. And now we have these tools, we have these ways of starting to chart the biological landscape even further. So it's an extremely exciting time I think, to be a biologist, to be a natural products researcher. And like you said, my background is similar in that I'm very much interested in medicinal plants broadly, and cannabis is a member of that, group of that focus of research. But I'm very interested to see what insights we gain by applying these tools and methodologies to all sorts of natural plants. And certainly psychedelic plants and fungi are becoming sort of the hype right now, especially like in where I live in Oregon in the United States. We just legalized the medical use of psilocybin this year, which is we're the first state in the country to do that. And so we are stepping into sort of a new world, because we're now going to have some models to start to collect patient data, to start to do chemical analyses, which I'm very interested in, on all of these different, I'll just say psilocybin products, because like you said, there's all these different species, and then within species, you have all these strains of fungi that have psilocybin and other psilocin and all these other compounds in them. But then diving into this complexity, what compounds, when presented together, provide the most efficacious result for depression or PTSD, or whatever. So, yeah, I mean, I anybody listening, I really try to impress just how special of a time we're living in. And honestly, just going back to how impressed I am with your lab and the models that you've made. I mean, this is something that in my 10, 15, 20-year plan, I hope to try to build something akin to what you've been doing, but try to get this happening in states like Oregon, where now we have the ability not just to study cannabis, but to study psilocybin products. We also in Oregon just decriminalized all drugs, and so that also changes what we can do on a research side. And that's going to continue to play out. So trying to figure out ways to take the insights that groups like yours have developed and say, how can we replicate those kinds of models? How can we, try to institute similar programs and then start to work towards international collaboration and sharing these data sets and start to really, I guess, see the bigger picture around a lot of these natural products. That's a big thing that's a big thing that's on my mind. To sort of change gears just a little bit, because before we wrap up, there was another paper that you published, I think this year, about transcriptomes of cannabis strains. And I wanted to ask you about that because when I've presented that paper to other people, sometimes people are like, why do we care about mRNA, and what does this mean? What's the sort of context for how this is going to affect medical cannabis in the future? So to kind of change gears completely here, I wanted to just ask you if you could briefly describe, one, what you learned from that research of looking at the transcriptomes of these different strains of cannabis, but also why is that interesting and important, and what are the implications as this sort of transcriptome research continues and you understand on a sort of genetic level more of what's going on with these strains?
So understanding the pathways, how the plant create the cannabinoids and which cannabinoids, what are the pathways, give us tools to, first of all, to learn in early stages of the plant, okay? And then how to change things that will be better. And so we are doing that with plants, with all the plants for years. You want plants that are growing in most, that will be stronger, less affected to pesticides, and other things. And so we're trying to make, first of all, the plants better, to be stronger, to give more products, to grow faster, and all of this. Mm-hmm. But in cannabis, we're not talking about just the tomato to make more tomatoes and it will be a little bit sweeter and redder and nicer and to have more products. We're talking about another fact, that the combination of the compounds are important to the medicinal effect.
Right.
And understanding the pathways that the plant produce these compounds are extremely important. Just a moment.
Yeah, no worries.
It's extremely important, and it's extremely important in order to create, to repeat the treatment and to repeat the grow and to change. So if we think that CBG is extremely important and we want to strain with CBG, how are we creating a strain now that having 16% CBG but very low amount of THC and CBD? because it can affect that. If we learn that a molecule 3318 is extremely important to leukemia, and now we need a higher amount of that, how we cause the plant to produce more. If you know the genetics, if you know what are the pathways they're producing, what are the enzymes, what are they, you can start to look for that first of all, and second, maybe to change it. So one of them is screening. You get a tool to screen just a moment. If I have this protein, if I have this enzyme, if I have this one, this mRNA, this plant will be like that. You don't need to grow that for 3 months and then to take flowers and then to check. You can, as a seedling, check that.
Yeah.
And this is extremely important because you can do things in large way. This is one side. The other side, you can create plants by breeding, by genetic, by CRISPR. So, if somebody don't like GMO, I can understand that. There is other ways. You can just do the right breeding.
Right.
You understand the mechanism. You said, okay, this plant doesn't have almost THC because this pathways is blocked because this mutation. If I will mate that with this plant, I will get this pathway here and that pathway here.
Yeah.
This is breeding, okay? But this is smart breeding when you know what you're doing and not blind, okay? So this is the importance of that. My lab is less dealing with that, even though this is my PhD. I know how to do that. But I'm usually working with the growers or breeders and or companies to help them to first of all define the molecules, the active molecules, to analyze it for them, and second to think with them together what will be the things that will be needed, what is interesting, what are
Right.
they. So I will tell you, I saw I got from one of the breeders in Israel a plant, a unique plant. It was kind of a mutation for him that having I just said that 17 18% CBG, zero THC, zero CBD. And when we ran that, we were fascinated to see that you can now describe what exactly the pathways, like which arms are creating, what are the THC family, what is the CBD family, and what is not. Because all the cannabinoids and terpenoids that are part of the pathways that creating THC and then downstream sub cannabinoids are just disappear. And you can see literally said, oh, there is just 9 cannabinoids left. And these are the cannabinoids that totally not related to this pathway, that pathway. So there is something that is, it's not just, there is a very, very specific reasons why to do that in order to make the plant better, in order to make the combination, the entourage effect better with a smart way, not as a blind. Yeah. This is science.
Yeah, and so you can basically create these mRNA profiles for different strains and partner that data with other tools to look at what's going on with the plant and everything, so that you can essentially make very targeted, informed decisions all the way upstream to the point of choosing which genetics you're actually going to cultivate, as well as being able to understand just the synthetic pathway mechanism. It reminds me almost the CBG example. It almost makes me think of like how researchers have studied just pharmacological activity of cannabinoid receptors and like doing knockouts in order to see, well, what else is still at play. Okay, well now we understand, that
this is very important to this, but not for that. It's fascinating. Again, it's not the bread and butter of my lab. I have a kind of cell biology animal model lab, and I'm not a grower. But because my background is from there, it's something that I'm being attracted to and be part of.
Yeah.
And we can help on that a lot, and that's what we're doing. Um, I think in this field of breeding, creating a new combination strain, again, we are just starting,
Yeah.
to understand what can be done. I think there will be quite soon other plants that are harboring cannabinoids,
Hmm. Yeah.
and this is a tricky thing because they're kind of legal. You
Yeah.
have all the phytocannabinoids inside, it's just not cannabis. I saw
Yeah.
the last year twice. One of them extremely interesting with the unique cannabinoids inside, cannabinoids that I didn't see in the cannabis, but also the regular ones. And I'm not talking about, I was familiar with 8 different plants that harboring cannabinoids,
Hmm.
but in a very low amount,
Yeah. Yeah.
0.001 CBD. Here we're talking about plants that have percentage of the,
Mm.
not in the
Hmm.
flowers, in the leaves, so you can get bigger biomass. And this is another thing that going, I think, to surprise us quite soon. It
Yeah.
can change. In Israel, the law said cannabis and its all parts.
Hmm.
Okay, this is the law. Cannabis on all parts and under the medicinal, it's illegal, recreational. It doesn't say cannabinoids. If I want to buy
Yeah.
synthetic cannabinoid, produce it in years time, it's legal. So if now you have a plant that have cannabinoids, but it's not cannabis, it's totally legal,
Yeah.
which is a tricky business here. Which,
Yes.
yeah,
Well, and this touches on what I was mentioning earlier, that I'm really fascinated to see how the concept of cannabinoids in general evolves, because that term cannabinoid, it has sort of a muddy history now, because it's sort of came about because of trying to identify the active compounds in cannabis. And so the cannabinoid term obviously relates to cannabis, and then that term gets applied to the receptors that Hallett's team found, and then later. So it's but now when you look at these plants that like, I can't remember the species, but that rhododendron plant that has a compound that's very, very similar in structure to CBD, with some very sort of minor modifications. And it brings up this dilemma of, like, when we see compounds like that are so structurally similar to phytocannabinoids that we're accustomed to seeing in cannabis, are those compounds cannabinoids or not? And then you get into this muddy concept that you sort of alluded to earlier, the endocannabinoid system, and really the endocannabinoidome. If, like, well, okay, we can start to see, do these compounds affect cannabinoid receptors? Well, even if they don't, that doesn't mean that they are not affecting endocannabinoid, signaling pathways, or other things. Maybe they're interacting with TRPV1 receptors or PPARs, or the myriad of other receptors that are being included in the endocannabinoidome now. Or maybe they're altering the biosynthesis of N-acyl ethanolamines or something. There's all of these different aspects now of ways to think about what is a cannabinoid, what are the functions. So yeah, I'm very, very interested to start looking into other plants with this sort of new perspective, and to look at compounds that we've identified in other plants that have been well known for a long time. I mean, a lot of these fatty acid derived compounds in the body that we've known about for a long time, like, now we're seeing them in a different way. And I see that taking place across natural products science in general. So it's, yeah, I totally agree. I think as we continue down this road, we're going to see more and more plants that have, if we don't call them cannabinoids, we'll definitely call them cannabinoid like. And this concept of the entourage effect, I think, is going to get muddier and muddier, because all of this stuff is interconnected. You can't, like you were saying at the very beginning of the episode, and maybe this is a good way to wrap things up, you really can't tease these things out in isolation. They all are connected to other systems. And so it's just funny the way we use these words: endocannabinoid system, cannabinoid, cannabinoid receptors. And when you really start to dive into it and really submerge yourself in the complexity of these molecular interactions, these words almost start to become meaningless, because you start thinking more about the actions and not really so much about the labels that we place upon the molecule or the receptor. And so it's it's a fascinating future of cannabinoid science, to say the least. That's ahead of us.
I agree. I think you said about the people that you are teaching, kids or students, that's feeling that they are missing the train. I think we didn't finish to put the railroad yet.
Yes.
We're just starting to understand that we're starting to understand that we want to get to the journey. We didn't even start to put the railroad. So there is a lot to do, and it's fascinating. And somebody asked me in a conference a year ago, isn't it frustrating me to work in a field that so much unknown? I said, no, this is my job. This is my occupation. That if it's known, I'm not there. I'm not doing the research.
Yeah.
This is science. This is research. And so I think it's so much unknown, and their potential is so big and so fascinating that we are lucky to be part of that.
Absolutely. Absolutely. And with that, that's a great place for us to start to wrap up here. Generally, the way I like to close out my episodes is I like to pass off the platform to the guests to kind of give any last comments, as well as let folks that are listening know how to learn more about the work that both of your labs are doing, as well as any presentations and things you have coming up. I know I think you're a speaker at CanMed again next year, and all of that sort of good stuff. So with all of this, I'll go ahead and hand over the platform to you. Let people know how to learn more and sort of what's on your horizons.
So first of all, thank you for getting me this platform to speak. It's always a pleasure. I think if people want to learn more today, it's very easy with computers and internet. You're writing my name, and there is the TED Talk and other things that we did. All the science that we're doing, we publish in scientific papers. So putting my name in PubMed, all the papers are coming out, but also in the site of the lab, Meiri Lab or Dedi Meiri Lab. My company called Cannasol, and they have also a site with all of the data, the details, the papers, what we're doing. I believe that the job of a scientist is to create data and give it away. That's why I'm getting salaries. So the data that I'm producing is when I really believe in that and I prove that it's real and I can put my hand on that, I usually publish that. Whether it's in a scientific paper, whether it's in our site, whether it's in my company site, we're not hiding anything. One of the first things that I told in my company in the beginning when we started, they coming all from companies and business and everything is to hide and not to translate.
You're right. Yeah.
If you want to lead, if you want to be the best one, if you want to run fast, first of all, you need to collaborate with everybody. Second, if you will be in the place that you're afraid that somebody will, steal from you and run faster than you, so you shouldn't be here. If you feel that you are doing the best thing, the best work, the best, so you shouldn't be afraid of anyone. Just,
That's excellent advice.
That's my message to my workers. Be faster, better, more accurate than everybody else, and then shouldn't care about what other people do. Just lead. And for that reason, I'm saying that because almost everything is there and easy to find and to read. I think I think science in general it's a puzzle. If you're a very good scientist, you're putting one piece of the puzzle. If you're an amazing scientist, crazy Nobel Prize, you put few pieces of the puzzle. If you really want to see the picture, you need to connect that the dots together. And then, what the Marzo is doing in the last years is phenomenal. And what the other scientists are starting to do in this field is phenomenal. And we need to connect these dots together and to work together as much as we can. And I must say that in this field, there is a lot of passion and unique people. And I didn't face till today somebody that didn't want to collaborate, to share, to do, I had something that I wanted my student to learn. I called Di Marzo. We didn't spoke before that. I said, hey, said, hey, okay, I read your stuff. And my student just fly to Quebec for 3 months and stayed in his lab to learn the method that he developed.
Wow. Yeah.
So this is one example, where we're all sharing the data, we're all sharing the methods, we're trying to help each other. It's a newborn field, and we need to work together here. And that's it. I'm open to everything, to collaboration. If somebody have any question, I'm quite busy, unfortunately, with a lot of emails and a lot of that, but I'm trying to be nice and open and collaborative, and everything that we have, we are sharing. So if some of the audience want to come to do a PhD, a postdoc, to learn in the lab, to spend time in Israel, to be part of what we're doing, if somebody who need help in his project out there, somebody need any help or collaboration, we are here. That's what we're doing.
That's beautiful. That's most excellent. And on that note too, knowing how busy you are, I can't articulate how appreciative I am that you've been willing to chat with me for over 2 hours here. It's been really great, and I've enjoyed the conversation just as much as I knew I would. Like I said, I've been wanting to talk to you for a long time, so this has been really great for me on multiple levels, not just a professional level, but also a personal level. Especially, before we talked, I didn't realize how extensive of a background you had running the medicinal plant gardens and all of that, which just makes me appreciate you even more, because I connect with that on a very personal level. So this has been very, 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 very awesome for me, and I hope that everyone that's listening, if you have been listening, you should have learned quite a lot, because there was a lot of information shared here. Some of it kind of sprinkled in and hidden, but if you have the right ears, you'll hear some really great things in here. So I hope this has been valuable to all of you listening. If you've made it the whole 2 hours, that's amazing. God bless you for making it the whole way. But with that, everyone that's listening, go look at Dr. Meiri's work. Like you said, you can go on PubMed, Google Scholar, whatever, type in his name, and you'll find tons and tons of papers. And if you want to learn more about Curious About Cannabis and the educational work that we're doing, you can visit our website, cacpodcast.com, or check us out on social media, Facebook, Instagram, or Twitter. And with that, everyone listening, stay curious and take it easy. Bye-bye.
Thank you very much. Bye-bye.
If you want to learn more about cannabis, check out the Curious About Cannabis book on Amazon.com and other major online book retailers.